Now that we've gotten a chance to discuss the specific larger structures within a cell, such as organelles within eukaryotic cells or cytoplasm found in all cells, let's continue our journey of looking within a single cell and explore how different components of the cell are truly interconnected to allow the cell to survive and thrive. Specifically, we will be exploring how proteins, aka the doers of the cell, are made from transcribing DNA into mRNA and then translating that mRNA into functional proteins. And then those proteins can carry out their functions throughout the cell. These processes are what allow life to persist, and they are constantly taking place within your cells too, ensuring that your body continues to function so you are able to complete this lesson. So thank you, cells. And with that, let's dive in. At this point, we know the basic structure of DNA and we know where to find it. Let's now dive into its function and how understanding this can help us better understand the connectedness of different components within a cell. By this point, I'm sure you have heard something along the lines of that DNA is what makes you, you, right? It's the reason your eyes are blue, or your hair is black, or you're somewhat tall. While DNA does contribute to these features, how does a line of code made up of A's, T's, C's, and G's cause this? It's actually a bit more complicated than most let on. Actually, DNA is just cellular instructions on how to make certain proteins. The proteins are what that contribute to your phenotype. Phenotype being your observable characteristics, right? Such as your eye color. So when people talk about gene expression or expressing DNA, right, what I really mean is that a gene within a DNA sequence was successfully used to make a functional protein. However, the DNA alone cannot express itself. DNA cannot do anything, right? It's just a set of coded directions. So the whole process of making a protein from the information from the DNA sequence is orchestrated by different proteins that participate in different parts of the process of gene expression or of expressing a gene. I like to think of this kind of like baking a cake in a bakery, right? To bake a cake, you have your cookbook, a specific recipe within that book, a baker, and your ingredients for baking. In this analogy, the whole cookbook represents a full strand of DNA, while a specific recipe within that book for a specific cake represents a gene. Genes are a length of DNA that code for a specific protein. The baker represents the ribosomes, which read the directions to make proteins. Now, just as a baker would want to make sure that their special cake recipe is preserved for many years to come, the cell also works to ensure that their DNA instructions do not degrade over time. This is where transcription comes in. Our baker friend takes the recipe from their cookbook and makes a photocopy of the recipe so they can use it in their messy kitchen without worrying. Similarly, for the directions of DNA to leave the nucleus, right, as though they were leaving the cookbook and into that messy kitchen, for the DNA directions to leave the nucleus, it is first copied or transcribed onto mRNA. Tying this back to Unit 0 material, do you remember any other reasons why DNA is transcribed into mRNA? So you can think of RNA polymerase in this analogy as the copying machine, right? And you'll see why in just a second. And then the ribosome reads the mRNA one codon at a time, as a baker would read the recipe one step at a time. Each codon corresponds to an amino acid, just like each step in a recipe refers to a specific ingredient. All right? Keeping in mind that the specific ingredient listed in the directions is not the ingredient itself, right? So in our directions, if it says get sugar, There isn't sugar physically in the cookbook. We just know that we should now go get the ingredient sugar because of what we read. Same thing in how codons correspond to amino acids. The amino acids are brought to the ribosome by tRNA, or transfer RNA. So you can think of these tRNA molecules like little interns helping out the baker. They deliver the ingredients to the baker, which in this case is delivering the amino acids to the ribosome. The ribosome then joins the amino acids together to make a polypeptide, just like a baker would mix ingredients together to bake a cake. So there's your analogy. Let's now go into a bit more detail on how this works within cells. So, for your edification, this sequence that we are discussing today is referred to as the central dogma. The central dogma is an explanation of the flow of genetic information within a biological system, specifically dictating that DNA is transcribed into RNA, which is then translated to make proteins.